Good afternoon. What better way to end up a Friday than by talking about the brain a little bit with a biochemistry lecture? So this will be like the old lectures, if you've been nostalgic for those. We're going to bring them back. But I will want to point out again, so you should be paying attention. This will be posted. You can actually go back and listen to certain parts of it. But I am going to quiz you on it in the way that we talked about it before. I'm going to ask you big questions. So what kind of big questions can I ask you? Now, I want you to still use big words to talk about those big questions and lots of details. So mention like what specific examples of a certain trait might be. But with all that said, I would just like to, this whole topic does lift out of the sequence of Lehninger, and it lifts into this course. So it's nice to be able to do a seminar about the brain today and about cancer on Wednesday. So let's talk about it. 3 parts today, 4 parts on Wednesday. And if we're talking about the brain, actually in common with all of the topics, in common with all of Chapter 12, is signaling of some sort. So what do we mean by signaling and what is going on with it? So let's move a far way away from brains, but let's look at one of our other topics, the immune system. One of the things that the immune system does, this is right here. It's neutrophils, one of those types of immune cells we didn't talk about very much, but they roam the body, okay? They're like, wasn't there that TV show Kung Fu with the guy like who would wander, David Carradine, and wouldn't he like wander the earth looking for rights to wrong or wrongs to right? It's been a long day. I'm going to get a lot of stuff wrong. But basically he wandered the earth like looking around for stuff to happen. Your immune cells are like that. They wander your body looking for stuff. For example, they can see laser damage in a mouse lymph node. And when that happens, if you light up all the neutrophils as green, you get a picture like this of what goes on. Here's the damage right here, and there they are moving towards it. They all, they know it's their job, it's their purpose to go after that laser and to do inflammation and see what's damaged and see what needs help. So that kind of movement, how do they know? They don't have eyes, they can't see. There's some kind of chemical signal that they are following that's causing a chemical change inside. And that's actually the whole topic of the chapter. There's something happening outside the cell that changes what's happening inside the cell. How does that work? It turns out that for very diverse processes, it's actually very similar. So here's another immune response. This one is kind of, I wonder if somebody has like put this to an audio at some point. This is another neutrophil, and it's going after this little bacterium. And so you see that the bacterium thinks it's getting away. It's not really, it's really randomly diffusing. Oh, it thinks I'll go this way, but then the neutrophil's right behind it. And the neutrophil, it's like, this is why in biology, and then, sorry, this is PG-13 for bacteria. So the bacterium does not end up in a good place. But the bacterium was caught and destroyed by the neutrophil. That is also chemically mediated. The neutrophil had to activate motion proteins that would move it toward the bacterium. They were good enough so that the bacterium could not get away. And by the way, that's as good as they have to be. They don't have to be better than that. They just have to be good enough to catch the bacterium. So what if there was not a bacterium, but a chemical? It turns out that they think they've found one of those chemicals that these are going after. This is slime molds, but they can move. And there's a pipette. You can see the tip of the pipette. And you can watch gradually, all the slime molds are like, hey, it's like a little feeder for the slime molds. But really what it is a pipette that is putting a particular chemical into solution. The particular chemical is a form of ATP. It's a cyclic AMP. Okay. So that's what you can do if you hold the pipette in one place. And it stands to reason that you could do this. I think this person was having a bad day at work or something like that. He just wanted to mess with the slime mold, because you can see that he moves the AMP over here, and the slime mold's like, okay, is it really going to be there? I've been burned before. And then move it over here, and the slime mold's like, oh, but it's not really thinking about it. Basically, yeah, eventually the slime mold is not ashamed. They can't feel shame. But you see how the response is happening in real time. There's some chemistry to that response. The signal moves, the inside of the cell moves in response. And actually, if you look at the chemistry of this, you can also get an idea of the time scale. What is the maximum fastest response you can get? Well, it's limited by diffusion. It's limited by the way bonds can form, break, and rotate. So you can actually talk about these kind of things and also work off some stress after a long day in the lab. Okay. The main thing is we went from complex to simple, but we have cyclic AMP, which is a signaling molecule. It's not providing energy, it's providing a signal. So that means it has a distinct shape, it's fitting into some kind of receptor, there's some kind of biochemistry going on. In this case, because the signal outside the cell is called the first messenger. It's sending a message, which is move over here to the different cells. And so we know there must be something that is both inside and outside. There must be parts of this process that move from the outside to the inside. It could be a single receptor embedded in the membrane, and maybe it binds to the AMP on the outside, and it changes something on the inside. Or it could be a transporter. It could actually let the AMP through. But that would be the same effect. It would be changing something on the inside by bringing the first messenger into the cell. And by the way, this is cyclic AMP right here. From remembering what AMP looks like and remembering your organic chemistry, you should see that phosphate can bind 2 alcohols. And if it can reach them, it can bind 2 alcohols on the same sugar. And so that's what it's doing. It's actually forming a phosphoester bond from the 5' to the 3' sugar. Very distinctive shape. And so you can make a receptor binding site that binds to that shape. And that's actually kind of trivial. That's like what we talked about with enzymes and things like that. Now, usually the first messenger can be anything that sends a message. So it can be even a peptide. It can be a protein, it can be a lipid, it can be, in a sense, the whole bacterium, although there's probably a simpler first messenger that the neutrophil was detecting as it was going. The first messenger is the first thing that hits the cell and tells it to send a signal of some sort. Usually, the only reason why this example, I couldn't resist using that slime mold video, but it's a little bit confusing because it uses as a first messenger what we usually see as a second messenger. It uses cyclic AMP as an external signal to the slime mold. Usually, inside our own cells, we see cAMP as an internal signal. So don't let that confuse you very much. Usually, we'll most often see cAMP used in our cells as a second messenger. So here are some first messengers. When we talk about immunity, antigens are first messengers. Infections. Growth factors, when we need to grow. Nutrients, odorants, and tastants. Okay? We're talking about the perception of the outside world through the 5 senses. Light is another sense. And neurotransmitters are also first signals. You know that you had the caffeine example that we were talking about. That was an example of caffeine acting as a first messenger, acting outside the cell with something on the surface of the cell, causing a change inside. You have a question?
Yeah, so in terms of like tastants, are they as comparably variable as odorants? I was actually talking to my girlfriend about this the other day. She's like, can you make anything taste like anything? Because for the most part, most things smell. Like, I don't know about that.
Yeah, it's a little more complicated. We'll touch on taste at the very end of the chapter. Because it is a certain kind of pathway, but it is a kind of signaling pathway. It's very similar to the odor pathway, but one of the important parts of it that we don't understand very well is there are external proteins that bind the tastes, and there's metals that interact with it. We actually don't understand the external part of it as well as we understand the internal. Therefore, I will talk about the internal part, but not so much about the external. But the things like that weird berry that changes your taste, I don't think we even know exactly how that works right now, but we have some ideas. And so open area of research, I'll throw it open to that. Mirko?
Just like neutrophil.
Okay.
What exactly is that? An immune cell?
Oh, yeah, it was an immune cell. I didn't emphasize it in the structural immunology lecture, but it was mentioned on the family tree very briefly. I'm not sure I even said it aloud. It's a form, it's like the macrophages. So for our purposes, they're all family members. They're all brothers and sisters in the immune tree of life, you know. So you can think of it as a macrophage, yeah. So, yeah, immunity is, I think it's sometimes deliberately confusing to keep people out of it because it's so cool otherwise. But anyways. So there are 5 general aspects of signaling that we'll try to use to organize our thinking. What does signaling have to be like to be a good signaling system for a cell? Well, first of all, it has to be specific. You want a particular molecule to bind a particular receptor and cause a particular result. By the way, these can be kind of fuzzy sometimes. Sometimes you can have a system that you want to be nonspecific in a certain way, but it has to be nonspecific in a specific kind of way. So we'll leave it at specific for now. Then you want amplification. You want a very small signal, maybe even as small as a photon, which technically doesn't even have mass. You want a small signal to result in a big change inside the organism. You want it to be modular. Interchangeable parts mean that evolution can take one signaling system inside the cell and apply it to lots of things outside the cell. And it looks exactly like that's happened. I used to think that we would have, like in biochemistry, you know, when we got to the real stuff in biochemistry, we'd have like a day for sight, a day for taste, a day for smell. No, they're all the same. And it's all a half lecture for those 3 because they're so much the same that the important parts are, you're talking about one, you're talking about the other one. They are modular. And that means that actually I can show you some experiments where scientists have used this modularity to build cool things out of yeast. But that's another story. That will come up. I've got to keep you, interested. I got to tell you, I don't have the big grade to hang over your head. This is just a quiz, right? But this is about the edification of knowledge, all right? And finding out cool stuff that scientists did. The other thing about signaling is you don't just want the amplification of the signal. You don't just want it turned on. You want to be able to turn it off. And that means desensitization, or, kind of a confusing word here, adaptation. Adaptation, in this case, is adapting to the environment like your eyes adapting to a dark room, or your eyes adapting when you walk out of a movie theater into a bright summer day. Okay, so adaptation in this sense is about turning stuff off to adapt to new circumstances so that you can receive another signal. Zach?
How paper are these conditions versus drug design?
We'll talk a few things about drug design because what we're going after signaling systems with drug design. We'll talk about RAS in particular, and you have that connection to Wells drug design. And these are all involving, drugs are first messengers. So these are all applicable, yeah. And we'll talk about that as it goes through. The last thing is integration, because you can have more than one signal. You're in a complex environment, and sometimes you want a case where 2 signals will add together. So 2 signaling pathways need to be able to talk to each other or overlap. Or you want to have them cancel out. Maybe you, like if you have the one signal, you want to turn it off when you see another signal. You want AND gates and OR gates if you want to think about it from a computer science kind of perspective. So here's pictures for each of these. Specificity is like, again, it's like what we talked about with enzymes and things like that. So actually some of the concepts from Michaelis-Menten kinetics and stuff like that, those actually can apply to this. We won't get into those weeds here, but I just want you to see how something fits in, how a drug fits into its receptor would be an example of specificity. Side effects, in many cases, are cases where the drug chemical is non-specific in a bad way. It's binding to something you don't want it to bind to. And that's one of the things we're trying to minimize, right? So here's this one, which is the next one. This is amplification. Okay, but you see that this is a device that is designed to amplify sound waves. so that a small little guitar string bending right there by just a few centimeters can be amplified by the pickup in the guitar, amplified by the tubes or whatever it is in the Marshall stack here, and it can basically make a really loud sound. Huge amplification that you want, and this is signaling music. We want to do this with chemicals. The way this is done with chemicals is through a cascade, usually. It usually looks something like this. So the thing about enzymes is they aren't stuck to each other. Once the enzymes can work quickly and they can work repeatedly. So that means that one enzyme can work on multiple substrates. That's the definition of an enzyme. It's not changed by the reaction itself. And so that means even if you have a very slow enzyme that can only interact with 3 other substrates, if that substrate is an enzyme, which itself can interact with 3 other substrates, then you can end up with multiplying your signal by 3, multiplying that signal by 3, meaning 9, 27, 81, all the way down. So you can have multiple levels of enzymes that can each operate on multiple substrates of the next enzyme, and that is an automatic cascade amplification. By the way, the green triangle here shows the cell's most popular way of amplifying signals, which is to attach a phosphate to it. You know, phosphates can go on pretty easily. They stay on for a long time. You can take them off pretty easily if you have the right site. And so this is a phosphorylase, or a kinase is the more common word used for it. You see this all over the place. We'll show you many examples of that. Modularity means you have interchangeable parts so that you can have a signal to a response with modular parts here. Even the phosphate can be considered an example of modularity, because you can just take the phosphate processing machinery, phosphate cutting machinery, or phosphate adding machinery, and you can put that in another protein context, change the binding site but not change the machinery, and you have a new signaling situation. Okay, so the whole idea of signaling actually there's a lot of interchangeable parts, and we will see things where there are shared aspects to many of these biosignaling pathways. We have a huge number of first messengers. We only have a few second messengers, and we only have a few examples of different kinds of kinase activity, and in fact, they're mostly kinase activity. It's just different protein interfaces is what makes them different. And so it really looks like these things have evolved from one to each other, with evolution using the parts interchangeably. Your eye looks like that, and it's possibly the most well-designed and well-honed system in your body. So it's the idea of interchangeable parts, and the idea of phosphates being able to be put on and put off, and enzymes being able to evolve into other ones. Okay. Oh, so there's this one. This one I want to show you. Stare at the dot. Don't ask any questions, just stare at the dot. Does anybody notice anything kind of funny happening around the edges of the dot? Yeah, now it's not animated. It's just staying the same. All of what you're seeing is what's happening in your eye. But what I see, and it depends on whether you move your eye or not, but what I see, this might work better on a computer screen, I sort of see the white closing in. When, so it looks like my eye is turning off the outer, you're not using that neuron anymore, so let's turn it off. It's kind of like you're staring at this one space, and it's desensitizing. So this is the adaptation desensitization that we're talking about. What this will look like in a signaling pathway is you will have some kind of signal being turned on, and Lehninger uses a green triangle for turning it on. But then you'll have, at some point, after it's on for a while, something else will go around and will turn it off, and will turn off the response with a red X. Sorry for anybody who's like my friend who is colorblind. He's using green and red just like stoplights, but he is at least using different shapes, so you can distinguish that. Green means go, red means stop. This is one I want to see if it works for you. I want to see if you observe, this is another desensitization, but you might observe something relative to color. pink dots. Anybody see another color? Yeah. And green, and then it disappears, right? Eh,
So mine is red. Camouflage.
Okay, that is a Mirko-specific phenomenon. Although it's also, you also.
Shows that
Wh-
everything goes around it.
Oh, okay. I think it has to do with the oh yeah, yeah, yeah. That's the animation. I think so that's real actually. But in a sense, the green is not real, or is it real? Because we're all seeing it, you know, except for colorblind people, I suppose. The green is not really there. The green is a processing artifact of desensitization, but it shows you something about how color is processed by your brain differently. And there's some relationship between the magenta and how you describe the green that you see. What's that? Okay, so like an exit sign or turquoise? Yeah, so not jungle green, Crayola green, you know, not the good green, not this green. Y'all know my favorite color. The one thing I want you to take away from biochemistry, I like green, so. But I like this green more than that green, but it's a different green. That has a particular relationship in wavelength, actually, and it shows you something about how your color receptors work. But again, we'll get to that. And the other thing is integration. I don't have a great thing for this. Integration is when you have two signals, although actually one of the upcoming, I have another illusion that I can show you, and it might be considered to show integration, where you have two signals coming in and one of them interferes with the other, either bringing up or bringing down the other. For example, if you have a receptor that increases a certain second messenger inside the cell, increases the concentration, you have a totally different receptor that detects a different signal but decreases the concentration of the same thing. They fight it out. And you know, the different enzymes, it depends on the KMs and the Vmaxes of the enzyme and stuff like that. But they can increase or decrease the concentration, and the concentration is one pool of the same concentration of the same molecule. And so depending on which signal is stronger, they'll be integrated at the point of the concentration of X. You can also do this with voltages. Remember that we have, if we change the concentration of a charged ion, that changes the voltage across the membrane. That's going to come back here. The one thing that you don't have to do is you don't have to do the complex homework that was with Chapter 12, unless you really want to. If you want to, just let me know. All it takes is one person. I'll just tell everyone who it was. But it's no, don't worry, no homework. Okay, so here's some more optical illusions that have to do with the processing of the neuronal signals. Like for example, like just stare at this one. Again, this is not moving, but do you sort of feel like it is? Your brain's trying to make sense of it, and your brain has a grouping function. It has an integrating function between neurons. But this is designed to mess with that function. And so, yeah, isn't it, it's kind of like, it's like looking at like the Cthulhu monster that causes madness or something like that. You feel like if you look at this too long, you'll start to see things and stuff like that. So here I just covered it up. Again, no animation, but you can see that it's not that puzzling when we just look at part of it. But if you look at the whole thing, you're like, it seems like it's just, yeah. Yeah. It's just a circle. But it doesn't look like a circle, does it? Okay. Okay, here's this one. Okay, I'm going to start this one again because I want to do it with you. This is my favorite. Stare at the dot. So you're ready for the dot. You stare at the dot and try not to move your eyes, because you'll find out if you move your eyes even a smidgen, it won't work. But we'll do it a couple times. The dot's up there. I hope this works. Okay, see the color? Now blink. Or move your eyes, and the color goes away. There was never any color. And you can look at it, you can confirm it, like if you don't look at the dot and then look immediately at it, or like you don't stare at the dot, like I'll be from the side here. And yeah, no effect, no color. Yeah, but it's a black and white image. It's another thing with color desensitization. So I have to do it one more time, see if I can do it. If you blink, it like messes it up too. And so I'm like trying not to blink. It's like being in a staring contest. Yeah, so you can try these. These might be easier on a small screen too, depending on things. So the main thing is those all show different aspects of signaling. We will eventually talk about light sending a signal, but that will be mostly in another lecture. So CAMP is a first receptor, and the way I think it works, I'm not 100% sure. It could be a transporter, but I think it's going to bind. And if I had to guess, I'd guess that it binds a serpentine receptor in the slime mold's membrane. The way that most life works, and hopefully slime molds are advanced enough to have this structure, everything advanced enough to have this structure will do most of its signaling like this with 7 helix crossing membrane structures, some kind of binding domain on the outside, some kind of signaling domain on the inside. Okay? The fact that there's 7 transmembrane helices is just one of those things you see over and over. And the question is this somehow optimal? Or is this just somehow locked in? Has this been like the first person to figure it out, the first slime mold to figure it out? Had 7 receptors. Can you get more or less? It's really interesting because evolution can do a lot of things, but doesn't really get away from 7. I've heard some arguments that it actually is optimal in some way. But it still seems oddly specific. But the important thing is this is how the signal gets inside the cell, some kind of conformational change that causes a twist that activates some kind of enzymatic kinase activity or some other kind of activity inside the cell, structural change. Once you're inside the cell, then your job is to spread the signal throughout the cell and start to make the changes that you want to make, like moving things around. Okay. So like, for example, this says move toward the CAMP. So there are 4 general types of receptors that will look like this in general. The G protein coupled receptors are the 7 helix spanning membrane ones that it will be. And again, all of these pretty much look the same. They also look modular in a certain way. This little cartoon applies to all of them. So there's some external ligand. It binds to a receptor, and the receptor binds what's called a G protein because it binds GTP. That activates an enzyme that will make a second messenger, and once you get to the second messenger part, then that can flood the cell. You know, second messenger, commonly CATP, small molecule, floods the cell and turns on a lot of other stuff. There's 4 other kinds that we'll be talking about. There's tyrosine kinases, which don't have a G protein on the inside. What they have is they just have some kind of dimerization event that will bring domains together and activate a kinase cascade in that way. Then there's some that directly produce a second messenger. These directly produce cGMP as a second messenger that will flood the cell and then go on. So, you know, why have something else go and make your second messenger? Why not just make it yourself? And yet this is less modular, and so it actually seems to be less prevalent than the more complicated but more modular approach. And then finally, you can just let the signal in. Instead of changing the shape of the protein, just open a door for it and let the first, or actually technically, the ligand here opening the channel is the first messenger, but it's letting the second messenger flow into the cell. Instead of making it, it's letting it flow in. That's the main thing there. So to just point out the second messengers in all these cases, you have a second messenger here, second messenger here. The ion or the voltage is sort of a second messenger there. And there's not really a second messenger in the kinase cascade necessarily, at least as shown here. So you realize that second messenger takes many different forms. And that is how things get started. So let's move on to the brain. I will